ICMR INDIAB study, post-field activities. Now let's look at the post-field blood sample processing activities. These consist of centrifugation, packaging and transportation of blood samples. Centrifugation. The purpose of centrifugation is to obtain serum from the samples collected from the subjects. Once centrifugation is done, the gel in the yellow tube comes in between the serum and the red cells. The centrifuge is an apparatus used in the lab for separating substances of different densities or particle size when suspended in a fluid by spinning them about a central axis in a suitable compartment at a given speed. We shall require the following to carry out this procedure: centrifuge, forceps, micropipette, micropipette tips, cryo vials, cryo box. Please note that only the samples from the yellow-capped vacutainer tube need to be centrifuged. Samples from the lavender-capped tubes should not be centrifuged. Centrifuge should be placed at a convenient location at the field site on a rigid flat surface. The centrifuge should be placed adjacent to a power supply and plugged in and kept on prior to beginning the procedure. Ensure that the rotors are clean before each centrifugation process and that the vacutainer tubes are tightly capped and placed vertically in the slots provided for them. Further, care must be taken to make sure that the tubes are diagonally opposite to each other and that the rotors are balanced at all times. If a balancing tube is required, a vacutainer filled with water can be used. Close the lid and begin centrifugation. Gradually increase the speed until 4000 rpm is achieved. Centrifuge the samples for 10 minutes at 4000 rpm. On completion of the runtime, gradually decrease the speed and bring it to 0, then switch the machine off. Make sure that the rotors come completely to the rest position at the end of the run before opening the lid of the chamber. Remove all the vacutainer tubes from the centrifuge using the forceps without disturbing the sample in the vacutainer. Place the sample tubes on the thermocol tube rack. Do's for centrifugation. Ensure samples are balanced in the rotor. Only equally balanced tubes must be placed diagonally opposite to each other. If necessary, an empty vacutainer tube must be filled with water and kept diagonally opposite for balancing. Close the centrifuge lid properly. Clean the rotor chamber once a week. Wear protective gloves and lab coat while handling the centrifuge. All the field workers must be familiar with the proper handling of the centrifuge. If one is unsure of how to operate the instrument safely, please check with a quality supervisor. In case of spillage in the centrifuge or during transfer, wipe off immediately with disinfectant. Please ensure that the centrifugation is done within 1 hour of collection of blood samples. Don'ts for centrifugation. Do not run the speed at less than 4000 rpm. Never attempt to open the lid of the centrifuge while the rotor is in motion. Do not lean or keep any object on the instrument while operating. Some anticipated problems and their solutions when carrying out centrifugation. If the vacutainers break due to improper loading of the centrifuge, then the centrifuge load must be diagonally balanced by placing vacutainers containing an equal volume of water opposite to each other. If the centrifuge malfunctions or breaks down, then immediately bring it to the notice of the quality supervisor, who will make the necessary arrangements. Aliquoting and labeling. Now let us see how to aliquot and label the yellow-capped vacutainer. Once centrifugation of the yellow tube has been done, the serum would have separated out and appears as a yellow-colored liquid. The serum is found at the top of the tube and the gel separates the serum from the red cells formed at the bottom of the tube. The serum is now ready to be transferred to cryo vials. Label the cryo vials and keep them uncapped for sample transfer. The serum is then transferred to 3 1.2 ml cryo vials. The transferring of the serum samples into the cryo vials should be done with the help of micropipettes. About 0.5 ml of the serum samples should be pipetted and transferred to each of the 3 cryo vials. This is the micropipette and these are the pipette tips. The micropipette has a fixed volume to draw 0.5 ml. Hence, no adjustment is required and at one time it will draw only 0.5 ml. To aliquot the samples, first the pipette tip has to be fixed to the micropipette as shown. This is the plunger. Then the plunger of the micropipette is pressed and inserted into the yellow tube containing the serum samples. To draw the sample into the pipette tip, the plunger of the micropipette has to be released gently. Once the sample enters into the tip, it has to be transferred to the 1.2 ml cryo wire by again pressing the micropipette. This process should be followed while transferring samples into all the 3 cryo vials. Now let us see how to aliquot and label the lavender-capped vacutainer tube containing the whole blood sample. The lavender tube has to be uncapped and aliquoted into 3 1.2 ml cryo vials with the help of a micropipette. Screw the caps securely onto the cryo vials and store them into the cryo box provided for them. Recap the used vacutainers and discard them into the bag provided for biological waste. The number of aliquots to be made for each type of vacutainer tube is presented in the table. 5 ml plain yellow cap vacutainer. 4000 rpm for 10 minutes. Number of aliquots 3. Volume of the sample aliquot 0.5 ml. 3 ml K2 EDTA lavender cap vacutainer. Centrifuge not required. Number of aliquots 3. Volumes of sample aliquot 0.5 ml. Dose for aliquoting. Transfer the serum and whole blood from the vacutainer tubes into the respective cryo vials. Write clearly on the labels. Take care that the sample does not lyse because of shaking or rough handling. Don'ts for aliquoting. Do not overwrite on the labels. Do not send the sample without sealing the labels with sellotape. Do not interchange labeled cryo vials. Do not create a vacuum with the tip of the micropipette while aliquoting the sample. Do not reuse the microtips. Some anticipated problems and measures to prevent when carrying out aliquoting. If the sample spills, wipe off and then ensure that the caps of vials are tight and intact. Please try to avoid wastage of sample. The entire procedure of aliquoting and labeling should be done carefully. If the writing on the label is smudged, then rewrite using the specified pen. Storage of blood samples. The next step is to ensure that the samples are preserved in the appropriate condition till they reach the lab for analysis. Following centrifugation and aliquoting, the samples are placed in the field ice box until they are transferred to a refrigerator prior to transport to the central laboratory. The items required for storage of samples are ice box, ice packs or gel packs, or dry ice and cryovial storage box. Arrange all the aliquots in the cryovial storage box. Now place the dry ice in the bottom of the ice box. Carefully place the vial box inside the ice box on top of the dry ice. Arrange the dry ice on all four sides of the vial box as well as on top of the vial box. Now close the ice box and tightly secure the knob of the ice box. Care should be taken to always hold the ice box upright throughout the transfer of the samples. Do's for storage. Make sure that there are sufficient ice packs or dry ice in the box before packing. Always seal the sample vials with sellotape before transporting. Keep the vials straight up. Always ensure that the temperature in the ice box is between 2 to 8 degrees centigrade. Don'ts for storage. Do not spill the samples. Do not keep the ice box open at any time. Do not keep anything else other than the blood samples in the ice box. Do not leave the ice box unattended in the field. Some anticipated problems and measures to prevent when carrying out storage. If the sample spills, then seal the vials properly with sellotape. If you find that the temperature in the ice box is not within the required range, then try to collect fresh samples from all the subjects before leaving the PSU. Please try to avoid this. Packaging and transport of all forms and blood samples to MDRF. Next, we will deal with the procedure involved in sample packing for shipment. Materials required for packaging are as follows: address label, thermometer, brown tape, thermocol box and dry ice. Prior to transport, the thermocol box needs to be within the labeled packing box and lined with dry ice. The address label should contain the following particulars: the printed address of MDRF, phone numbers with area code of MDRF and the instruction to handle with care. The address labels should also contain the printed label, biological samples on it. The samples in cryovial box are then to be placed within the dry ice lined thermocol box. Dry ice is then placed on top and sides of the samples. The core temperature of the box should be then determined prior to sealing the box. The temperature of the box must be maintained between 2 to 8 degrees centigrade at all times. Document the temperature of the box and seal the box tightly with brown tape. Pack the box in a disposable cover and seal it with tape so that the cooling is maintained. Then place the details of the collected samples in a Ziploc cover and place it inside the package to be couriered. In addition, place the completed questionnaires, white and blue, location map, structure map, log sheets and ECG rolls in separate folders, which are packed separately as paper documents. Pack them properly, seal with sellotape and courier them along with blood samples. Enquire with the courier service about the time taken for the parcels to reach MDRF and inform the MDRF staff of the same. Do's for packaging. Make sure that there is sufficient dry ice in the box before packing. Always seal the sample vials with sellotape before transporting. Ensure that the temperature in the box is maintained between 2 to 8 degrees centigrade at all times. Don'ts for packaging. Do not spill the samples. Do not forget the labels. Do not forget to document temperature. Some anticipated problems and their solutions when carrying out packaging. If there is a courier strike, then store the samples in a freezer until the strike is over or until you find another courier. In an extreme situation, you may send the box to MDRF with one of the field staff. Waste disposal. This session of the video deals with disposing the biomedical waste. The biomedical waste includes used vacutainers, used needles, used pipette tips, used glucose meter strips, used lancets, used cotton, and used gloves. The biomedical waste should be segregated as per the specified guidelines. All biomedical waste should be segregated according to their type and drop them in the respective color-coded bags only. There are 3 color-coded bags. The green color bag. It's for disposing paper and plastic items such as disposable cups, plastic waste, food, glucose waste, etc. Blue colored bag. For disposing needles, lancets and sharps. Red color bag. For disposing infected bandages, vacutainer tubes, glucose meter strips, infected cotton, swabs and gloves. After the disposal in the respective color-coded bags, the bags should be closed tightly and taken to the local lab where the samples will be analyzed. The waste disposal bags should then be handed over to the quality supervisor or manager, who will hand them over to the state PI for disposal. Care should be taken not to leave any of these biomedical wastes in the field.